I'm going to talk about very newer concepts in terms of targeting the ubiquitin proteasome system. As all of us know that proteasome inhibitors are big successful agents in the treatment of multiple myeloma. However, most patients eventually develop resistance to proteasome inhibitor, and that this therapy is also associated with toxicities and off-target sites. So in order to overcome this issue, we have started and focused on examining ubiquitin proteasome system, in particular upstream of 20S proteasomes. And interestingly, we found that the 19S lid of the proteasome harbors not only the deubiquitylating enzymes, but also ubiquitin receptors. So our studies have focused on really delineating the role of DUBs as well as receptor. I will give you a couple of examples. We started working on two major DUBs which are linked to 19S proteasome lid, which are USP14 and UCHL5. And what we have is a proof of concept drug. And these enzymes basically function by removing the ubiquitin from the substrate and thus allowing the entry of the protein into the 20S core proteasome, where the catalytic activities of the core works and degrades all the proteins. However, if you block these DUBs upstream of 20S proteasomes, you basically block the whole proteasome mediated protein degradation. And one such example of a drug which landed in the clinic, and it's the first in human DUB inhibitors. Similarly, we also have ubiquitin receptors, which are the first recipient of ubiquitinated substrates. They recognize the ubiquitin substrates, and then together in cooperation with ATPases as well as with DUBs, RPN11, they allow the protein to be prepared or primed for the entry into the 20S proteasome, where it gets degraded. So we have recently worked on one of the ubiquitin receptors called RPN13, and we have utilized a drug, proof of concept drug, to target RPN13. And we have shown that once you block this ubiquitin receptor, there's a huge accumulation of polyubiquitinated substrates, and thereby showing that the proteasome mediated protein degradation is blocked. And in fact, it is much pronounced than proteasome inhibitor like bortezomib. Similarly, we have RPN10 as a ubiquitin receptors, and we are doing extensive drug screening against the RPN10, and we have some initial data sets which are not presented in this meeting yet to show that we have blockade of RPN10 also induces complete proteasome mediated degradation of the protein. So this is very exciting because we now know that the resistance to the proteasome inhibitor could be in part due to mutations in the catalytic subunits of the proteasome. And now we have a way that we can go upstream of the 20S proteasome and block the proteasome completely. So, and can overcome any mutational mediated resistance to the proteasome inhibitors. In addition, one interesting fact is that RPN10 not only works for proteasome mediated protein degradation, but it is also part of autophagy, or we call it a second protein degradation pathway through the autophagy and lysosomal pathway. And we have recently found that blocking RPN10 can in fact take care of both proteasome mediated as well as autophagy mediated protein degradation pathways. These together would allow for much more enhanced inhibition of all the degradation of the proteins. And these are all basic science research, and we still need to have much more work done on this issue. But nonetheless, it is important to let this thing out to the research groups to understand, as well as to the clinicians, that we now have a way of harnessing the ubiquitin proteasome system in a much better way. Another example of such aspect is targeting the ubiquitin specific protease 7, USP7. We published in Cancer Cell in 2012 that this is the DUB that can also, independent of proteasome, can also trigger cytotoxicity in multiple myeloma. And therefore, we have gone on to make a drug against this USP7 in-house, and in collaboration with Dr. Sarah Burlug and structural biologist Serrano, we have now a drug that is undergoing clinical testing in animals for PK and PD studies, and hopefully we will have another DUB inhibitor which is targeting USP7 and for the clinic in the future. So these are some exciting times of how we are going to overcome the proteasome mediated resistance.